Okay, this is David Zierler, director of the Caltech Heritage Project. It's Friday, October 20th, 2023. It is great to be here with Dr. David Stauffer. Dave, it's great to be with you. Thank you so much for joining me.
Thanks for inviting me.
Dave,
This should be fun.
good. Dave, to start, would you tell me, please, any titles and institutional affiliations you currently have?
I am a senior consultant with CGI. CGI is an acronym that formerly stood for Consultants to Government and Industry.
What kinds of things does CGI do?
CGI has consultants all around the globe, and I would say we are involved in all manner of industries. We've got different markets that we serve. I know we're strong with financial services. We're strong with government services. My particular involvement is more on the life sciences side of things. Prior to being employed by CGI, I was with a smaller company called Paragon. And so when Paragon merged with CGI about 5 years ago, I was part of that merger. And I've been involved in pharma IT as a consultant / business analyst for about 12 years now.
Who are some of CGI's clients? Are you working both in a government and a private industry space?
I'm just in the private industry space, the commercial space, as they call it. So the clients are any of the big pharma that you might know about, whether it's Pfizer or GSK or Merck. I've worked on projects with many different large pharma, and so I would say that we're fairly active in across the pharmaceutical industry, at least in terms of the branch of CGI that I'm most familiar with.
Dave, what kind of expertise do you bring to these partnerships? When you're a consultant for industry, what are they looking for from you?
They're looking for mainly, I would say, CGI is mainly IT consulting, a lot of strategic consulting. So what they're looking for from me is help with managing the data related to discovery, related to commercialization and manufacturing, so that that data can be related to chemistry, related to biology, related to all of the processes that it takes a pharmaceutical company to go from identifying candidates for therapies to characterizing them, clinical trials, bringing them to the manufacturing shop floor, and in order to take your drug and make it ready to sell to patients.
Do you get to be a scientist at all in this role? Have you transferred to data science or even systems engineering? Or how is your Caltech experience, your expertise, relevant in what you're doing now?
That's a great question. You asked, am I a scientist? I'm not a bench scientist in the sense that I'm not the chemist, I'm not the biochemist or biologist who is trying to discover or otherwise optimize the manufacture of small molecule drugs or biologics or vaccines. So I've kind of jumped over to the IT side of things. But where my Caltech background comes in, particularly in Dennis Doherty's lab, I was in the lab making molecules with Dennis, but there were others in his group that were doing computing. This was 30 plus years ago, so the computing back then was a lot different than what you could do today. So I would say the arc of my career has taken a lot of the different things that I've learned from Caltech and allowed me to bring them forward to different aspects of my career. Mainly, I see myself as something of a translator for science and IT. That is, scientists in the lab are trying to find new therapies or optimize the production of those therapies. And I'm able to help them understand what IT tools are available to help to serve them. And when it comes to building IT tools, I know who the people are that are going to be using them and what their requirements are. So that has kind of placed me in the role of a generic term business analyst. So I can help the business, the scientists, communicate with those who are going to be building them systems, IT systems mainly.
Dave, have you always been a computer guy, a data guy, or is that sort of a pivot after graduate school?
I think that was something that started in graduate school. It perhaps started earlier, and we can come to some of that. But it's kind of run in parallel. In undergraduate, in my experience as an undergraduate, that was mainly focused on hands-on work in the lab as a bench chemist. Similarly, when I was at Caltech, my graduate project was oriented to what we could characterize with molecules synthesized in the lab. And as a postdoc, I was also doing laboratory stuff. But running in parallel, there's been, hey, do you know how to use these computer systems or computational chemistry tools? My first job after my postdoc was for a small company called Cash Scientific. And they had some really cool technology for visualizing molecules in 3 dimensions. And that was appealing to research chemists. And so they needed to learn how to use these tools. How am I going to do computational chemistry and 3D visualization of molecules? So I was part of a team that was introducing those tools to biotech, pharma, sometimes even agrichem, anywhere discovery and seeing what your molecules look like can be of value.
Dave, what have been some of the most important technological advances, ranging from computation to instrumentation, even machine learning and an artificial intelligence, that really influence what you're doing today?
I'm not sure of any particular technological advances that have influenced the direction that I've gone. I've done a lot of work with sort of the high-end workstations, 25, 30 years ago, computational chemistry workstations. So in terms of technological advances, being able to take what were really specialist tools and have those become more commonly available, I think has been a benefit to science. Although while that was happening, I was moving from computational chemistry into information systems more generally, the management of chemistry and biology data related to drug discovery. And so going from the kind of standalone workstation systems to more of a client-server architecture, that is, those workstation systems now became powerful enough to connect over networks to a large number of users, so that you didn't have necessarily only the power user I thought of myself as a power user, and in talking to power users, I was also talking to more, I guess, I don't want to say general purpose scientists, but those scientists that don't routinely go to a computational chemistry workstation and say, I want to understand how to dock molecules. The tools did evolve so that from a client-server standpoint, managing data became something that the bench chemist could do on their own. And so rather than computational chemistry, it's, hey, I'm making a series of molecules. I need to share what I'm doing from my bench, whether it's through an electronic laboratory notebook, which is something that came relatively more recently, past 15 or 20 years, whereas 20 to 30 years ago, the client server architecture was being developed to serve the information management side of things and being able to register molecules, register reactions. The one company that I worked with, MDL Information Systems, way back in 1978, they pioneered the workstation reaction management system. They actually had a reaction management system before they had a molecule access system. So they were pioneering, how do we represent a chemical structure both graphically on a screen and at the same time have it as a set of information that we can understand computationally, informationally, so that when scientists need to find out about the molecules they're working on and the relationships to molecules that they can buy or that they can compare to what's known across the industry or later on across the internet, they have the ability to search and find any bit of information that's available about molecules of interest to them, reactions that can make those molecules, biological properties, chemical properties, and the like. So as I'm walking through this, I realized that, yes, there's been an evolution of technology that's been part of my career arc, going from that kind of standalone workstation model, which evolved to client server. Now you've got cloud-based, where the servers are, they're no longer inside your institution, whether it's a university or a pharmaceutical company. So that cloud-based architecture now makes it so, hey, I've got a browser, I can do the same types of knowledge mining that I used to rely on whatever I had available inside my institution or what I had here on my workstation. So now it's a wide open environment where the tools have matured, or I would say are still maturing, to enable that basic question that the scientist has, what's known about this this therapeutic area, what's known about biologics that are targeting oncology, what's known about small molecule drugs that are antiviral agents that can benefit a wide variety of patients. So I'll pause there because I've...
No, that's great. That's great. You alluded to it, but is your area of expertise then relevant across the broad spectrum of drug discovery and delivery? You're not just focusing on oncology or neurology or pulmonology. It's everything. If it's medicine and it's human health, what you offer is relevant.
Yeah, I would say so, and remind you that it's more from an IT standpoint, where there are tools and systems that um, make it so that all of those niche therapeutic areas are readily supported, so that when scientists have questions they need to answer or information that they need to capture to share with their colleagues, share with their companies, they have readily at hand the ability to do that.
So that means that from an IT perspective, it's all information, it all is relevant to whatever the drug is that you're talking about.
In a broad sense, yes. You've used the term data science, and I see myself more of a data scientist than a business analyst, although that's not to disparage business analysts, but my background as a scientist, I can speak the languages of chemistry, biochemistry, biology, molecular biology. So, and the kind of the languages of information systems. So that terminology I use as a translator is brought to bear. I'm not interacting with scientists and saying, have you ever considered trying to do this with your experiments? Mine's more of understanding that here, these are scientists that want to be able to optimize the conditions for purifying the manufacturing process related to a key biologic for oncology.
Dave, because your clients are competitors, how do you deal with information that's proprietary? What firewalls do you have to make sure that your work with one company doesn't spill over in terms of what you know in working with another company?
That's a great question. So when I'm working with a client, I'm largely, while I'm not an employee of the client, I'm a consultant to the client or a contractor, I am in effect embedded and treated as though I am an employee in terms of I have access to the client's IT systems. I have a client laptop that I work with so that I can fully engage with the project teams and any of the other stakeholders that we come across, whether they're scientists or people in regulatory or clinical areas besides manufacturing or discovery that we might come across that there's in terms of firewalls, in effect, by using the client laptop, I'm behind their firewall. I'm using their virtual private network. I'm using, I have access to their systems to get, like any other, let's say, at Caltech, you have a laptop, you have different kinds of training that you go through to make you eligible to have access to certain systems. So with each of the clients that I work with, I'm getting for a given project, I'll get a kind of a similar baseline training with each client that does all of the the human resources stuff, but and all of the onboarding that is needed to shall we say make me eligible. Now, can I get to the scientific information systems and do the training there that make me effective as a consultant?
Dave, how much of your work is on site? What can you do remotely?
That's evolved over time. Early on, you'd have to be on site to do all of the work. And then as remote work became more prominent through the 2000s, I've been able to work primarily remotely up to the pandemic. Since the pandemic, it's been it was exclusively remote. Now, I can go back to the client sites as needed. It depends on the nature of the project. I've been with one, I would say, a rolling set of projects that have involved a lot of the same team members. And so having, as a consultant, the familiarity and the credibility with them, I can engage with them and meet with them virtually, like you and I are meeting, using the appropriate intranet tools for the client. So, short answer to your question, right now I'm almost fully, exclusively remote. I'm available to go to the client site as needed.
Well, Dave, let's now take it back, establish some personal history. What were you doing before you got to Caltech? What was your area of focus? What opportunities presented themselves that got you to Caltech?
Well, before Caltech, I was at Penn State. I got undergraduate degrees there. I did a bachelor's in pre-medicine, a bachelor's in chemistry. There was something they had called a simultaneous degree program that you do all of the requirements for each degree, and then you have to have a certain number of credits above and beyond. I was able to pad my credits by placement exams for Spanish. I had Spanish middle school, high school, and I placed out of 12 credits worth of Spanish, which, more than 40 years ago, excuse me. You think about how much credits cost today, and that was, those placement exams are worthwhile investments for me. But I also was doing research. I started doing undergraduate research as a sophomore with my organic chemistry professor at Penn State's York campus, a gentleman named Ernie Harrison. He got me started in the lab. He also got me aware of being able to take graduate level courses as an undergrad. So when I went up to the main campus, University Park, I was taking graduate courses, and I started working in the lab with Phil Deschamps, who was an assistant professor there. I believe Phil is probably by now emeritus at the University of Maryland. But in any case, getting into the lab, synthesizing molecules, learning safety in the lab, all of that was great foundational stuff to be doing, so that when I got to Caltech, it's like, okay, I have some idea of what I can be doing here.
Dave, being on, let me just ask, being on the pre-med track, were you already thinking about the possible connections from chemistry to biology? Was that sort of already on your major even as an undergraduate?
Yeah, perhaps not in those terms, but yeah, I was looking at, having an impact in medicine. I got excited with organic chemistry and saw, hey, here's a way that you can make molecules, and of course you understand that drug discovery involves making molecules. At least that was the primary way back then. So whether it's making the next big molecule that's going to help a lot of people, or being a practitioner of medicine in practice or in a hospital, those were, I would say, none of those were fully formed, or even there was no formation of those ideas back then. But I was looking at, trying to make a difference through science, whether it's chemistry or biology. I took, in addition to graduate level chemistry courses, I took some graduate level biochemistry courses. I think for the pre-med degree, I had to do, that was embryology. Embryology almost, that was the most challenging biology course I had at Penn State. And that was in part because the wet labs that we had to do, they were a little messy is not the right term, but you get the idea that when in the chemistry lab, there's a lot of clean, neat, organized stuff you have control over. When you are doing biology, your containers, your environment of study is, moving toward living creatures.
Life is messy.
Yes, that's a very good reminder. Another thing that happened at Penn State that had me involved with computing, I took Comp Sci 101 and did punch cards, but that was good to have done. But I was fortunate to be involved with a group of other undergraduates who were in chemistry, biochemistry, bright, enthusiastic, and looking to do some interesting things. One of those, I'm guessing by now you've interviewed Steve Mayo.
Haven't gotten to Steve yet.
Okay.
I will, though.
Give him my regards.
I will.
We're both Penn State alums. And while, I'll let him tell his story of Penn State, but I was aware that Steve was doing some really cool things with computer graphics. And when he got to Caltech, and he and Bill Goddard and Barry Ellison formed Biodesign, they needed somebody to put their modeling algorithms to the test for small molecules. And so by the time I was at Caltech and in Dennis's group, I knew some molecular mechanics. I was familiar with those tools and how to use them. And so Biodesign asked me if I could do some consulting for them. So I would spend a few hours a week helping them to characterize how their molecular modeling package did with small molecules. They were focused on proteins and larger molecules.
Dave, how did Caltech get on your radar? Did you know about Dennis Doherty even from Penn State?
I did not know about Dennis Doherty from Penn State. Caltech got on my radar as, one of the top chemistry graduate schools in the country. And they also have a strong biology department. So there was the sense that, you had asked me about the pre-med, and I did apply to MD, PhD programs. I got accepted to none of them. I applied to 7 graduate schools for chemistry, and I got into all of them. So,
what is the takeaway there?
Well, the takeaway, I think, was that my undergraduate, arc shifted from kind of biology, medicine to chemistry, organic chemistry, perhaps bioorganic chemistry. In terms of how I appeared as an MD, PhD candidate, they probably saw strong PhD, MD. We don't know if that's, where he belongs. and you decide where to go to graduate school, and I had envisioned I was going to do synthetic chemistry, synthetic organic chemistry, and as you're deciding where to go to graduate school, you make a visit to the campus. I remember visiting, visited Harvard and MIT, and it was in the middle of winter, and there was snow up there. And when I visited the West Coast, visited Berkeley and Caltech on the same trip. I think they also combined that with a trip to UC San Diego. But in any case, you get out to Caltech and it's like late January or late February, and it's beautiful. So that got your attention. But more importantly, when I was interviewing and meeting professors in chemistry and biochemistry and biology, I got introduced to Dennis. He was put on my schedule, even though I didn't know anything about him at the time. I was aware of Dave Evans and Bob Ireland, who did synthetic organic chemistry. I had heard of Peter Dervin. And I had heard of some of the folks on the inorganic chemistry side. I can recall there were 7 of us from Penn State who had applied to all of these schools, and 7 got accepted to Caltech. I know 6 of us went there. Another one went to Berkeley. But I remember Harry Gray from, the inorganic side of things. He came to Penn State. He gave a talk, and after his talk, we got to go out to meet him. He wanted to meet all of these students that were looking to go to Caltech or another school for their graduate studies. And Harry makes an impression on everyone. He certainly made an impression on us. But he helped us start to see that the Caltech environment is, you don't get pinned down to any one thing.
Right.
It's wide open. And that was exciting for me to know that I was going to have an opportunity to do graduate studies with some really top-notch people. And when I'm interviewing, I meet Dennis Doherty. He tells me a little bit about what he's doing. He's like, "Oh, that sounds neat. Never knew he was doing that." He was relatively new at the time compared to his more established senior colleagues. I believe he was an assistant professor and was about to graduate his first PhD student, Lisa McElroy White. But I remembered about Dennis is that, he's originally from Harrisburg, Pennsylvania. I'm originally from York, Pennsylvania. We're like 20 minutes away. He had gone to Bucknell. I went to Penn State. I knew, so I got to know a bit of his biography. But in terms of deciding to come to Caltech, even though the synthetic organic chemists, I believe Dave Evans moved on to Harvard before I even arrived at Caltech. And within a couple of years, Bob Ireland would go to pursue another opportunity. It's like, well, gee, synthetic organic chemistry, I guess I'm not going to be doing that at Caltech. Should I consider going to another school? And it's like, no, there's enough stuff wide open there that's going to challenge me and feed me. So stuck with the decision to go to Caltech. You arrive, I think what seemed strange to me was um, everybody else is getting into graduate school Labor Day or a day or two after, and it was like the third week of September till we come out for the start of the fall semester or term or whatever it was in fall of 1983. But by then I was thinking Peter Dervin or Dennis Doherty, because Peter was doing some really cool stuff too.
Right.
And when you get to Caltech, okay, you have to do some coursework, and you've got other graduate students now. We're all jockeying for position of, okay, whose lab here, whose lab there, this and that. And I think there ended up from our class being 3 who joined Dennis's group. There were probably 4 or 5 who went to Peter Dervin's group. And the thing that was appealing to me about Dennis was he had some, to me, really innovative ideas. And his background as, he'd done some theoretical calculations, computational stuff. His approach was more mechanistic organic chemistry than and it aligned well with what I had experienced as an undergraduate taking the graduate level courses. Mm-hmm. It seemed like a good fit with the dynamic of the group. There were 2 guys in the area above me. The year before, Tim Chapad and Mike Petty had started, and they were doing the synthetic host guest chemistry. They were kind of working on the first prototype. I was kind of coming along, and it looked like I'd be doing the second one, a different class of structures to try and characterize synthetic host guest models and understand how, the simple naive question was, how do you bind grease in water? And so you needed to have a greasy inside and a water-soluble outside. So even before you emailed me, probably about a week or 2 before, I had put this together. I have the models that we used in the lab at Caltech. The CPK models are much better in terms of giving you an idea of what you're building. So this is a molecular model kit that I've had around for 30 plus years. And I thought, well, do I have enough pieces to be able to build what we had? Not exactly, but it's close. And so kind of the design and build of these, this is the first prototype, the Petty Chapad prototype, if you will. The one that I did, I don't have that model yet. What we ended up finding, of course, is that you can bind a greasy thing inside the host, kind of a donut shape, and characterize a lot of things about them. I feel like I need to pull back and help you reorient me to your questions, since I think I've gone way off track.
No. So, I mean, right now I can get to the major research question at hand. In considering Caltech, or once you got your bearings at Caltech, had you come across the term chemical biology? I know at this point, the mid and late 1980s, this is really the revolution that Peter Dervin and others are leading. But was the term already being used, or is this a term that's sort of applied retroactively in recognition of what the new changes were at this point?
I guess for me, it might be the latter. Um, I don't know that we used the term chemical biology when I was at Caltech. There were those of us in the division of chemistry and chemical engineering. There were those in the division of biology. When I was meeting people to decide on which graduate school and then which lab, you talk to people in different divisions, even though, hey, I'm in the division of chemistry and chemical engineering. But the term chemical biology, that came later. We had new labs that were built within the first year or two I was there. The Arnold and Mabel Beckman Laboratories of Chemical Synthesis. Interesting. I'm pretty sure that was built to support the professors who were doing chemical synthesis, who perhaps had moved on to different locales. Not that we weren't doing chemical synthesis, but that wasn't our main initiative. Or, Peter Dervin, Dennis Doherty, their approach is to try and understand, nature through chemistry tools. And chemical biology as a term that if they were using it then, I missed it.
So, Dave, what would have been the term then? Would it have been inorganic biochemistry? What would it have been?
I guess we called it bioorganic chemistry.
Bioorganic chemistry.
Yeah.
Okay.
So,
even though the ideas and the methodology were really different than traditionally what bioinorganic chemistry would have conveyed. Yeah. This is the newness of chemical biology, even if the term was not already in play.
Yeah. I suppose it's kind of jumping back a little bit. Leroy Hood was at Caltech, I believe, when I started, but after he was in Division of Biology, but he was already taking some of the inventions and the protein synthesizer, the DNA sequencing and synthesis equipment that they were building there and commercializing it. I believe Applied Biosystems was started probably maybe even a year or two before I got to Caltech. Former Hood graduate students and postdocs through that. So those became the kinds of tools that bioorganic chemists might use. Peter Dervin's doing the sequence recognition molecules and doing a lot of biomimetic. I would suppose biomimetic is another term that we used mimicking biology through chemistry, biomimetic chemistry, whether it's synthetic host guest chemistry or the things that Peter Dervin was doing with his group to great success. So the terminology was, I suppose we tried to stay buzzword compliant and consistent within our field. I think as a naive graduate student, you don't know that, oh yeah, you get to do this cool research because somebody's funding it.
Yeah.
And, so people like Dennis and Peter, they're getting grants approved that fund the graduate students, fund the postdocs, and that if you want to be like those people, you'll have to be writing for grants as well. Um, and so, how you characterize your field and your role in your field and what you're doing, there's an art to that level of communication that I think that now chemical biology is an effective term.
Right. Dave, this will invite obviously an imprecise answer, but as a graduate student, as you're emphasizing, breaking down these departmental, even these lab boundaries, did you feel in your group that you were all chemists who were now doing biology? Did you feel like it was a new science altogether? Did you feel really like you were doing biology from a chemistry perspective?
Not at that time. I would say, you mentioned kind of, I don't know if you said this, but breaking boundaries. As I think perhaps a fourth or fifth year graduate student, I sat in on a neurobiology course that was, one of the instructors was David Anderson. There was another instructor whose name I don't recall, but and this will help connect how I got from Caltech to Columbia for a postdoc.
Okay.
So I'm sitting in on this neurobiology course, and I'm learning about acetylcholine receptor. There was maybe 3 to 5 lectures that were around membrane neurotransmitter proteins, membrane-bound neurotransmitter proteins, and the prototype there is the acetylcholine receptor. And acetylcholine is a quaternary ammonium compound. This is not it, but this is a quaternary ammonium compound that we studied at great length in Dennis Doherty's group. And so we were learning that if you want to bind a quaternary ammonium compound, you might use aromatic structures, benzene, naphthalene, anthracene type structures, which is what we had in our synthetic host. In nature, that means aromatic residues such as phenylalanine, tyrosine, tryptophan. And so the neurobiology course, I started to dig into what's known about the binding site for acetylcholine in the nicotinic receptor, and found that, well, there appear to be some aromatic residues. How well characterized is that? And this got me thinking that if the principles that we're learning in Dennis's group in the synthetic host guest realm is mimicked in nature, we should be able to find ways to demonstrate that. So I actually asked David Anderson about, hey, who covers nicotinic acetylcholine receptor stuff? And he named a few names, including Caltech's Henry Lester. But by this time, I'm thinking, what am I going to do for a postdoc? And so I'm looking outside of Caltech. And I ended up going to Columbia to work with Arthur Carlin, who had spent a significant part of his career investigating the acetylcholine receptor. So I knew I was going to get to do more molecular biology, more biochemistry, broaden my set of tools, lab tools, to come back to, at Caltech, kind of auditing or sitting in on this neurobiology course gave me, I suppose, inspiration for some next steps in my career. And so having the latitude to do that was great. And I know since then that, Dennis and Henry Lester got together and they've collaborated to do a lot more in-depth characterization of the nicotinic receptor for acetylcholine, as well as other neurotransmitter membrane proteins. So we kind of, I suppose we ended up both being inspired and evolving in terms of what are we going to do that's going to be really cool and help us understand how nature works.
Dave, because you were at Caltech all over the place in a good way, how did that affect your putting together the dissertation? How interdisciplinary was it, would you say, as a result?
The dissertation was fairly straightforward. It was for my work with that I did in Dennis's lab. There are 3 proposals that one submits in conjunction with the dissertation. So one of those was related to the implications of the work with Dennis for the nicotinic acetylcholine receptor, kind of the stuff I was going to go off and explore. I don't recall the other 2, quite frankly, but the dissertation itself was, bring together the work that I did in his group, and we haven't talked in depth about what that was, but part of what was important wasn't that second prototype that I was working on. It became, looking back at the first prototype, I described how the synthetic host in water could bind grease. The synthetic host in an organic solvent could also bind this, which gave us some insight that the aromatic rings, and this predated the acetylcholine receptor stuff in terms of thinking that this should work in nature, but binding this quaternary ammonium compound in an organic solvent, that synthetic host has properties that don't require strictly what we would call hydrophobic binding. I fear the water. I go hide from the water, and the safe place is inside this greasy donut. So the greasy donut has properties that bind something that is, this quaternary ammonium compound is interesting. It's water soluble, but it's very greasy. It's got 1 nitrogen and 10, 11, 12, 13 carbons. But it's the atom mantle trimethylammonium, its acronym ATMA, the binding of it in an organic solvent told us the magnitude of binding that one could expect from a biological system for acetylcholine or for any other positively charged organic molecule. And that led to a lot of work down the road that reinforced that for not only acetylcholine receptor. There is a story to tell. It pertains to there's an enzyme called acetylcholinesterase that takes acetylcholine and splits it apart. And so in terms of acetylcholinesterase, where is that important? Your body has it. If you have I'm thinking of bug spray, and bugs get sprayed with the toxin, and acetylcholinesterase gets all bound up and it can't do its job. Bug dies. It can't process neural signals anymore. Basically, paralysis. A few minutes, they're done. So when I was at Columbia University, I believe it was my second year there, Dennis and I had submitted a paper to Science describing the molecular recognition of acetylcholine by a synthetic receptor. So synthetic receptor. Now receptor becomes the term that replaces, synthetic receptor instead of synthetic host. And one of the data points that I collected in my graduate work was, hey, can we bind acetylcholine with our synthetic receptor in water? Yes, we can. 10 micromolar, not as strong as the sub-micromolar binding that we had for something like this, but nevertheless, what it showed and what that paper helped others to recognize was that binding of organic molecules in an aromatic environment was possible, particularly positively charged small molecules. There's a friend of Arthur Carlin's at Columbia. I shouldn't say, I'll need to correct that. An investigator in Israel named Joel Sussman is an X-ray crystallographer, and one of his areas of study was acetylcholinesterase structure and function. And apparently in his group, they had a crystal structure that they were trying to understand the implications for. One of the people in his group, I believe a postdoc in his group, is a gentleman named Israel Silman. Israel Silman had been in Arthur Carlin's lab as I believe a graduate student. I recall Dennis and I had our publication in December of 1990 in Science. In January of 1991, Israel Silman was visiting Arthur Carlin's lab, and he came to meet me because the paper that Dennis and I had published had given them insight that this cavity of aromatic residues at the bottom of which sits the active site for acetylcholinesterase, it made perfect sense that there were, I recall the paper they published said 14 highly conserved aromatic residues that line the cavity to the active site for the acetylcholinesterase. And he wanted to thank me for, sharing or finding this out with the synthetic receptor because of its implications for their work. Now, to them, all of this made sense. They were thinking, this can't be right, until they understood that from our paper that you can have aromatic residues that are, in effect, the face of the aromatic residue is negatively charged. The edges are positively charged, partial charges, so that, this diffuse negative charge, if you've got a whole cavity of them, you're going to be able to pass this type of structure down through there. So that's another squirrel we've chased in this conversation. We'll be back.
Dave, why Columbia? What was so compelling about Columbia based on what you had accomplished at Caltech?
So what was compelling at Columbia was that I was going to have the opportunity to work with one of the key researchers for nicotinic acetylcholine receptor, Arthur Carlin. And when I had interviewed with him to see if I would postdoc, he also had me meet not only others in biochemistry, but he had me meet people doing molecular biology, basically all of the people that I need to work with to carry forth this idea that I was bringing to his lab. So that was exciting.
Now, to foreshadow a little bit here, did you already have interest in going into industry Were you going to a postdoc because you were all in on faculty opportunities eventually, or were you keeping sort of both avenues open?
Great question. I went there with the idea of going into academia.
Mm-hmm.
That was my objective. I knew that I might spend 3-4 years doing a postdoc. I was anticipating I would be getting a lot of experience with biochemistry, protein biochemistry, molecular biology, and be, adding to my knowledge-based tool set with the idea that I could attract chemists who are interested in solving biological problems, biochemists, biologists who weren't afraid to, think about things from a chemistry standpoint. I thought, there will be a lot to attract them. So I had interviewed for faculty positions. I would say in hindsight, what I didn't appreciate was this kind of trying to be a chemist doing biology or a biologist doing chemistry, is that your first faculty position, at least when they were looking at me, they're looking for someone who they can put a clear, I think label is probably the right term. Are you an organic chemist, or just what are you? And so I wasn't enough of a chemist for the chemistry departments or biochemist for the biochemistry departments. I in effect failed to market myself at it correctly or properly to recognize what is it that my customer wants.
Of course, this is a strategic problem for Caltech alumni because they're so far ahead of the field in the way that they define themselves. You need 5 or 10 years for the rest of the field to catch up. This is not the first time I've heard something like this.
Yeah. So I can't lay the blame at hey, the world didn't catch up to me, but I think I'll admit to being naive, and I've learned a few things since then, as we all do.
Dave, leaving Columbia, thinking about the job market, how had that shifted your perspective on what was happening at Caltech? Not from the inside looking around you, but from the outside looking back, in terms of particularly what Peter Dervin was doing to the field.
I thought that what Peter Dervin was doing to the field was validating something that I said naively while I was visiting Caltech, still an undergraduate. And what I said to one of the professors I interviewed with was, and this was a biology professor, I said, I don't think biologists are doing their research with enough of an understanding or taking into account enough of the chemistry that's involved. And that was the professor was very charitable in the way the response was made, to at least allow me the arrogant naivete to elaborate in terms of, as to come back to Peter Dervin, he's been teaching the field that, and those that have gone forth from his group have been teaching the field that you can do a whole lot of exploring of nature using chemistry.
Mm-hmm. Mm-hmm.
And if we call that chemical biology, sounds like a good fit to me. But, looking back at Caltech from when I was at Columbia, that was still kind of the phase of, I don't have what academia is looking for, and that kind of stings.
Mm.
So, circumstances have me looking at industry, because it wasn't like I was saying no to academia, but I needed to find something that was coming next, and that turned out to be working with a software company, doing computational chemistry, as mentioned, Cash Scientific, and then about a year and a half later, getting into managing information, chemistry, biology data, and being the translator. But in terms of what was going on at Caltech and how I hadn't been following the field as carefully as when I was a graduate student and a postdoc. So my kind of intellectual investment there had diminished.
Dave, just anecdotally, among your peers, both at Caltech and Columbia, was industry an increasingly viable pathway for careers at this point? With all of the advances, with the emergence of biotechnology? Was this something that was an easy pivot to you that might not have been had this been your experience 10 or 15 years earlier?
Yeah, certainly, I would say so. Thinking back, I would say, the area that became a chemical biology for those of us in the Doherty lab, the Durbin lab, similar labs, part of what was happening was that, biotech was starting to blossom, and they needed smart people, creative people who knew how to solve problems and use these very innovative tools, molecular biology, protein biochemistry, and the like. 10 years before, the maturity wasn't there. But I hadn't really thought of it as, turning to industry as a more viable option. I know, thinking back to the folks that I'm aware of from Caltech and Columbia, that those who are in academia have, in effect, figured out how to kind of grab onto their ideas and not let go, and pursue them through funding agencies. And at least for myself, going into industry has given me the latitude to solve different kinds of problems in creative ways that the funding comes from the company. So it's a job instead of I guess I don't know how it feels to be a principal investigator at a university who's renewing grants every few years, applying for new grants to support students. That model I didn't get to have the experience of, so it's for me foreign.
Dave, obviously it worked out in industry. It was not the same experience you had on the job market for academia. What aspects of your experience, both in graduate school and as a postdoc, particularly in IT and data management, computation, where was there that obvious connecting point as you pursued industry opportunities?
Um, the connecting points were um, something I alluded to earlier as a translator, being fluent in science, and uh, to that, and compared to others who would come into the industry, I was, for someone who spoke science well, I was pretty conversant in IT. And so going into industry, then it becomes, how do I speak IT better and better without losing the science? And so being in pharma IT helps that a lot. I would say, I'm not, I don't follow the cutting edge of synthetic host guest chemistry. I occasionally will peek back at, Dennis's list of publications and see, okay, what's come since. I know that there was a crystal structure that was published of just the extramembrane portion of the acetylcholine receptor, which included the binding site, which helped to confirm all of the notions that we had going in. I know Dennis and Henry Lester wrote I think a Nature note on the Nature paper, a note that came along with the paper that gave that structure from a lab somewhere around 2000. So,
Dave, in your first experiences out in the so-called real world, how much were you bringing where there was a demand because they didn't have it, industry was new to this, and what aspects were you joining something that was sort of already in building mode?
Okay, see if I understand.
Meaning, were you attractive in industry because there was this new combined expertise that you had and they needed it and didn't have it? Or was that infrastructure, was that already in progress and you were joining that?
Good question. I think it is more the former, that I was, when I joined MDL Information Systems after Cash Scientific, I was identified as someone to, hey, are you looking for a job? I had a job, somebody says, hey, we think you'd fit in well here, because of the ability to speak science and speak IT. And at least in my younger years, I was still getting comfortable speaking in front of audiences about both of those together. So there was, opportunity to grow with the job as far as, the position on my resume says field application scientist. That's basically a pre-sales. I'm going along with the sales guy as the subject matter expert on how the software works and serves the science that these folks are trying to do. But they recognize that I would be a good fit for their sales team, introducing what was, at the time, cutting edge client server chemical information management systems to pharma. And so for the next 3 to 5 years, we had, we were hitting our revenue targets and exceeding them tremendously. So what that means is you make your target and you get a bonus and everybody's happy. The salespeople are making big piles of money. Those of us in field applications, we're doing all right too. So it feels like, this industry thing isn't so bad.
What were the satisfactions just in comparing a life of basic research, whether or not it connects to applications, to really doing something where there's an obvious end goal in mind, both as applies to human health impacts and profit margins?
You asked that again? I was hearing the second half, but
Yeah.
I missed the front.
I mean, it's the basic divide between fundamental research, curiosity driven, without regard for what comes of it, versus being very end goal oriented with human health impacts and profits.
I would say that uh, I would argue it's not so black and white even in academia. There is a target. The basic research it's framed with a target in mind.
Mm-hmm.
And those of us with targets, to achieve those targets, there's still some kind of to try and come to your question, both of those there's basic investigation, creativity, activities that are involved. That if we have our pharmaceutical clients, they have markets that they're trying to serve. You've got every pharma company wanted 10, 15 years ago, it's the $1 billion molecule. Now it's the $20 billion molecule. I believe with COVID, that was the $90 billion molecule, as it were, for the Pfizer vaccine. But in any case, that thing that's going to pay the bills so that we can do the basic research in pharma that's going to find us the next big molecule or successful revenue generator so that we can continue to thrive as an organization and help our patients do the same. To come back to your question, I think finding the creativity, the basic research, kind of freewheeling aspects, there are aspects of what I do as a consultant that are innovative they're probably more innovative in terms of starting with, here's a problem to solve. What's the best way to solve it? I probably don't have the best way to solve it. I have a quick way, a way to solve it. It might be a quicker way than the best way. A lot of the consulting I do is project-based, so there's a temporariness to what needs to be done. Mm-hmm. So this is a kind of a long roundabout way of saying what I do, in fact, for a lot of my consulting project work is I live and breathe in Microsoft Excel. Because this grows out of having worked with an information systems company that had databases for chemists and biologists, databases that run on platforms like Oracle. And so I'm familiar with working with Oracle databases. I'm familiar with scientists preferring to work with Excel spreadsheets. And so providing them tools that can either allow them to work directly in Excel or in an Excel-like environment is often very appealing. The learning curve is shallower than if you have a new application that's nothing like they've ever seen before, but helping to solve problems with managing information that people can communicate with using Microsoft Excel is a lot of what I do the past 5 to 10 years.
Dave, I'll ask a macroeconomics question. Between, for example, the dot-com boom and bust at the turn of the century, the financial crisis, 2008, 2009, how does that affect your day-to-day in terms of stability, in terms of the science, in terms of the computation you're able to do?
I would say I think I have a sense of where to take this now. Kind of the economic impact and what does it mean. So big changes in the economy mean big changes in what clients are focused on, and mergers and acquisitions happen. And as a consultant, any kind of change is good because it means that people that weren't working together before now have to learn to work together. And for the people, and that also means that their data has to work together. So you're bringing together mergers and acquisitions model of, big pharma. Company A works with this kind of system. Company B works with this kind of system. We're either merging, we're acquiring. We need to have new systems. Even the systems that we had before are, I don't know that it's when it started becoming planned obsolescence, but it's well understood that IT systems don't last forever.
Mm-hmm.
And so newer ones come along that are better, cheaper, faster. We talked about going from that workstation, client, server. Now we've got the cloud. Somebody wants to go to the cloud. Well, what does that mean? What do they do with the information that they have already in these databases that they spent, time and effort and money on? And so as a consultant, I'm, from a strategic standpoint, advising on what the options are and what, the recommendation is for getting from where you are to where you want to go. And here are the things that you need to keep in mind, because it's going to take you 2, 3, 4, 5 years to do that. So when companies do due diligence on, a pharma merger, part of what they're doing is at least identifying what IT systems are impacted, so that they can be, their CIO can, of whatever the new company is going to be, can be making decisions about what are we going to keep, what are we going to make legacy and let sit, and what are the new things we're going to do.
Dave, a related question, you've alluded to it, but it should be asked more directly. You've lived through so many mergers. What does that mean in terms of appetite for risk-taking? Where is a merger good for pushing the science, pushing the data in new directions? And where does it really mandate a more conservative approach? And what's better?
This is a difficult question for me, because I would say, as a consultant, I see IT departments that are generally more risk averse. They don't want to be the reason that a pharmaceutical company doesn't make its target in terms of market. So if you think about the COVID vaccine, there was a lot invested in making that all happen, with the idea that the return would be there. And I believe that was very wise to do that. To the question about risk and the impact on mergers and acquisitions, I'm going to need you to pull me back again with the question.
That, when companies merge, because you've seen it from the inside, when one company acquires the other, just generally, what does that do to the new company's appetite for risk?
It tends to make them less tolerant of risk.
And is that generally not welcome by you? I mean, is that antithetical to pushing the science, to pushing data initiatives in new directions?
So with a merger, you've got staff from both companies that realize that change is coming, and company A, people know what company A systems are like. Company B, people know what company B systems are like. And so now they have to figure out together how they're going to play nice. And you're going to have some people at company A that go, I'm going to go do something else, whether it's with another company or what have you, take the golden parachute, depending upon where they are in their careers. Same for company B. So when you're trying to get the new systems to work together, there's generally a lot of aversion to risk, because it's like, I'm familiar with what I'm doing. I don't want to have to do, to take on what somebody else is doing. There are, this is sort of the people that are, shall we say, left on the island, mm-hmm. that haven't left or been voted off. They eventually recognize they have an opportunity to do the next new thing. And so they go from more risk averse to being ready to take risk again, because, they understand that change, once they accept that change is coming, if they can be in charge of that, then it's like, okay, I'm ready to take that risk. As a consultant, kind of watching this and making recommendations, recommendations are typically based upon what we've seen in other circumstances. No two things are exactly the same. So, we're sharing perspectives more in the abstract than the concrete. But because we as consultants get hired to help with the process, we're keeping an eye on not only what the new set of tools are going to be and what the new IT systems are going to do. We're involved with the term in the industry is the change management, and helping the people who are, still on the island to be able to adapt to the things that are new and the new things that are coming.
Dave, how does this inform your decision to change company? When do you stay put? When do you see the opportunity to move on?
I'm not sure that, at least with where my career is now, that applies. My employer is CGI, and so I am a contractor through CGI to different pharmaceutical companies. Are you asking about when I go from one pharma to another?
No, in your own employment, when you're a consultant, right. How does
okay.
Mergers and acquisitions, risk taking, how does that inform your decision making about when to stay and when to move on to the next opportunity?
I guess in my career, when I was with MDL Information Systems, I went through 6 mergers and acquisitions, and this is company sizes around 500 plus or minus a few hundred people. And it was the 7th one that I was part of a reduction in force. And so that was something that happened to me. It wasn't my choice. Through that, then I positioned myself in the job market as a business analyst, and I've been working as a consultant, but through an employer company. So to come back to your question and think about a move I made from Paragon to BT Global Services, that happened when the role of BT Global Services was positioned as something similar to what I had done as a field application scientist with MDL Information Systems. I was going to be involved in, teacher translator of sciences for information systems related to discovery research. And so I guess I found something. I was like, hey, I enjoy doing that. I'd like to try and do it again. That position turned out not to be as valid as I had anticipated it should have been. So happily, I hadn't burned bridges at the company I had left 7 months previous, and came back to them and was able to land on my feet, as it were, in a new project in a different area of the pharmaceutical industry.
David, embracing the business side of things and positioning yourself as a business analyst, obviously you don't have an MBA, you don't have a business background. How much of it is just common sense? How much of it is understanding the business side from all of this experience? And how much did you really need to read up on economics and corporate finance?
Good question. There's a lot of common sense that comes with experience, I would say. I think, part of what you get from Caltech, Columbia, the progression is there are lots of opportunities to learn as you go and to explore, there's a certain attitude of, I want to find out more about this and understand it better. So in terms of understanding business, it's recognizing that, hey, when I was in graduate school, I was working in the lab, I was trying to solve this problem that was leading me to, my dissertation. Whereas, the people that are using computational chemistry tools or information systems in industry, they have a different set of problems that they're trying to solve. Let me understand what those are so that the value proposition of our IT systems and tools is presented in a way that makes sense. So, there's the business of what do I need to do for my job, but also the recognition that this this ecosystem that is pharma IT, the pharmaceutical industry, that there are different objectives that different levels have. And as a field application scientist working with salespeople, we had every few years we get some refresher sales training. And so solution selling, customer-centric selling are effective communication models that are brought to bear to be able to understand what your client's overall business is and to start to see what particular people that you're interacting with, whether they're on the IT side or the science side, what their critical business issues are and how you can relate to them.
Dave, moving our conversation closer to the present, we already covered how COVID impacted you in terms of remote work. What about on the science side, on the biotech side? Did you have a front row seat to how some of these companies responded to the crisis, both in terms of just work culture and coming up with therapies?
I would say work culture, yes. On the therapy side, no, not in terms of the science. The work cultures, there's a general challenge of how do we keep the trains running? How do we keep manufacturing medicines when the people are not able to be on site? And so, this comes down to companies readily recognize that the people that are involved in manufacturing are essential. And so they developed the safety protocols with. I don't know that they necessarily had full hazmat suits, but I remember seeing the movie The Andromeda Strain when I was much younger.
Oh yeah.
And so kind of have that picture in mind when you hear hazmat, at least when I hear hazmat suit and biological contamination and emergencies go. But to your question, the work culture became one of, at least in the projects that I've been involved with, one of, how am I going to work from home? And it's not only, how do I do the work that I'm responsible for at home? Particularly, if somebody needed to be in the lab to do something, they were now, figuring out how to structure their workday to do that and to remain effective in their other aspects of their job. So, not everybody went remote. My role is one that could be remote. There were others whose roles could be remote, but they hadn't been remote before. And so the work culture is one where you have some people that don't have a dedicated home office and they have to try to create one. Maybe they have a spouse who's also in the similar boat. Where am I going to, be? We had, some cases where, you've got two working spouses. The daycare that their kids would go to, no longer available to them. So they're now, they're scrambling and trying to figure out, how am I going to get my work done? How am I going to manage all the family stuff that used to be managed in different ways? So I would say that, the first 3 to 6 months were very chaotic for lots of people. And my role was to not only to recognize that they're struggling, but to remind them that, hey, we can do this. Our projects have goals that, you're helping us meet by, we'll get it done in the 1 or 2 hours that you have available to be meeting with us in a given day. And, there became a new, the new meaning of work-life balance became, I would say it became life-work balance, because life suddenly became the dominant during the workday.
Right.
That we're helping people with.
Dave, we'll move the conversation right up to the present. What are you working on right now? What's important to you?
I work on making sure that the laboratory equipment and the computers that support that equipment are state of the art, that they are secure, and that the people that use them have all of the tools on those systems to do their work. And that work is primarily in manufacturing of biologics and vaccines. So mine is a support role. If any one of those systems goes down, there's a backup for it. I have experience with helping one of my clients to bring systems back online after a cybersecurity incident that wiped out half the systems in the company. And I was part of a team, and we were able to do it in 2 months when they thought it would take a year. So, and I was able to be helpful because of things that I had experience with going back to Caltech. It's like, hey, let's learn how to use this computer. Going back to Caltech, a silicon graphics workstation. Man, that was the greatest new toy. But even before that, there was the Macintosh 2CI, and then there was the IBM AT, the bread box. And so back then, there wasn't a whole lot of software for doing scientific work. So certainly the silicon graphics workstation did. But, you start, how do you learn to play how do you learn to use a computer? You probably started by playing some games. And then, okay, now we need to do some meaningful work, because games are fun, but you master them, then they become boring.
Well, Dave, hearkening back to what you learned at Caltech is the perfect segue to the last part of our talk. Now that we've worked right up to the present, let me ask first, how have you stayed close to the science? You've said, over the course of our discussion, you're fluent in science. Now that you're so involved in industry and IT and the data, is it just sort of like the language that you learned and you just stay on it? Do you keep up with the literature? Do you ever go to conferences? How do you stay close to all the science?
Good question. I would say it's more from having not lost it. How do I know I haven't lost it? I think I'm still an effective translator when I am in, the client laboratory space talking with the scientists, and they're talking about, hey, we need to take this cell culture and move it over here, and we're doing this and that in these conditions. And I know what they're saying, and I'll ask them a question or two, and they'll do this. You understood what I was saying? You know what we're talking about? Yeah, I do. And part of it comes just from sort of day to day, when you've got when anybody goes to the doctor or dentist or any other kind of medical stuff is going on in the family. I'm often invited to be along to make sure that we don't lose anything in translation, so that I can ask questions and help family members to make sure that they're understood. Because I know from talking to them, members of my family, that I unfortunately often talk over their heads, and it's not intentional. It happens. I go off on tangents as I have in this conversation, and it's like, get excited about something. It's like, I don't think they understand what I'm talking about. How can I make this simpler? So I have a constant reminder to keep it simpler. My kids are great at reminding me of that.
Dave, I'm after the elusive hunt for the term in the history, chemical biology, right? So we've already covered. This was really the term, not the science, but the term itself was not front and center to you either in graduate school or in your postdoc experience so much. What about over the course of your career? Where have you seen the term crop up? Where have you associated it with the advances that you were a part of at Caltech?
Let me think about that. Can we take a short break?
Yeah.
So I'll be right back.
You got it. And we're back. So Dave, the question was where you have encountered the term chemical biology, or even just the ideas behind it over the course of your career?
Yeah, I think the term is not something that I come across and that I've heard used in my roles either with MDL Information Systems or my subsequent second half of the career as a business analyst. But the concepts are, I think they're pretty much ingrained and have been ingrained in the pharmaceutical industry for over a decade. And particularly in terms of pharmaceutical industry going from kind of small molecule drug discovery to expanding to include biologics. So a lot of the medicines today are grown in cell culture rather than synthesized in a lab with solvents and flasks and so forth. So yeah, the term is not familiar, but the concepts have been around for over a decade, easily.
Dave, have you kept up with Caltech over the years? You keep up with Dennis or your fellow students?
Not as much as I would have liked. Let's see. I am aware of their different careers or roles through tools like LinkedIn. I think I mentioned earlier, I'll check out Dennis's Caltech page from time to time. See if see how far back I have to go till I recognize names. It's like, wow, I think I was last at Caltech in 1999. I happened to be back for there was, I forget exactly what it was, but there was an opportunity for me to travel, I think, on business and swing by for a few days, or maybe it was a vacation that I expanded into that. But I got to meet some of the people in their group, and then current grad students and postdocs, and it was a bit disarming because they I think viewed me as something of a legend, and when you're there, you're just a grad student trying to figure out a set of problems and make enough mistakes until you're successful.
Dave, of all the work that you've been involved in over the course of your career, what's been most satisfying, both in terms of, helping businesses achieve success, and then even beyond that, what it is that the business is ultimately after, which is developing drugs that help people?
So my post-Caltech career, when I was with MDL Information Systems, they were introducing a biological screening data management system, and it was kind of an empty canvas that companies could use. And what it lacked, at least what I saw that it lacked, and it was understood by others that it lacked, this was kind of a set of examples that showed you what this could be. And so I created a set of examples that were rich enough and wide-ranging enough so that the system, it was given value in the eyes of people who couldn't see value before. Mm-hmm. That was rewarding to be able to create something like that using the tools that we were going to be turning over to companies, and the people within those companies are going to be able to take what, shall we say, the paint by numbers and then go create their own works of art within the tool. Even though those works we're keeping track of all of the different levels of screening and testing that we're doing to determine which therapeutic candidates we're going to advance. So that's from the discovery side.
And then the business side, where have been some major business successes that you've been a part of?
There are the business successes that are, hey, we made our revenue target. Those are satisfying in the moment. It's like, hey, we did. It's like we won the game. But I think that it comes to, I mentioned earlier, this company that had a cybersecurity incident that needed to keep its medicines in production. And they were scrambling to make sure that their supplies remained intact. So I had a small part in helping that manufacturing process continued. This was something that was before the pandemic. I wish I could say during the pandemic something that I did made a difference with, one of the vaccines, but I was not involved in any of those.
Finally, Dave, I want to ask a question that looks to the future, because obviously there's so many more advances that we can imagine, both in data science, IT, and in pharmaceuticals. What do you see as having built as an enabling technology for the future? If you've ever served in a mentorship capacity, or you ever work with young people as they look to forge their careers, what have you built that might suggest where all of this is headed?
What have I built? I'm not sure I've built anything like that. I guess I think for someone I would mentor, it's to, I'll give you an example. I've told you about my facility using Microsoft Excel. Mm-hmm. And I can share what I do with someone, and they recognize that that's pretty cool. But until they have the curiosity to go dig in and go find something else to do with it, it's not theirs. Mm-hmm. And they're not going to carry it forward. It's still mine. So I would say to someone I would mentor that develop that curiosity to go figure out what needs to be figured out. And be relentless.
Dave, on that note, this is... No, go ahead.
But I was going to say that I've had numerous times with problem solving where my attitude of it begins with, this can be solved.
Yeah.
It might be really hard. I might not be the one that gets to do it. I've seen throughout my career other people have been able to do it, but the attitude is one that this can be solved. If I get to do it, great. If I don't, someone else does it, that's fine too. But we all have our contribution to make.
But believing that this can be solved, that's really the Caltech way, right?
Absolutely.
Dave, this has been a wonderful conversation. I want to thank you so much for spending the time.
Oh, thank you for the time.